When you hear the word virus, what comes to mind? Well, you probably think of infectious pathogenic agents such as these. Well, then it may surprise you to know that our genetic material is 8% made of viral genes. These are known as endogenic retroviruses, or ERVs for short. And these are a type of transposon. There are a lot of interesting things to learn about them, so let's not waste any more time. To begin to understand ERVs, let's look at how retroviral DNA becomes incorporated into host DNA. First, the virus attaches itself to the host cell and the viral envelope fuses with the host membrane. This causes the RNA from the retrovirus to be inserted into the host cell, along with key proteins such as reverse transcriptase, integrase, and protease. The first problem faced here is that in order to become incorporated into the DNA, RNA needs to be turned into DNA. This is done by reverse transcriptase, which is represented by the blue oval. This double-stranded DNA then enters the nucleus, where it will be incorporated into the host chromosomes. The enzyme integrase that is found in the viral body allows it to become incorporated. If you look to the right, shown is a viral DNA segment. The three main genes found are the GAG, PLO, and EMV, which are responsible for key aspects of a virus. The ends are capped by long terminal repeats, or LTRs. LTRs are essential for allowing the new DNA to become part of the chromosome. To understand how ERVs occur, let's consider some of their many benefits. The remains of viral DNA is actually useful to humans in a couple of ways. They help increase gene expression and also genetic variation. However, arguably the most interesting benefit that human ERVs provide has to do with the placenta. The placenta, as shown here, is the organ responsible for the circulation of waste products and nutrients between mother and the developing fetus. The umbilical cord is what allows for the nutrients from the mother to reach the fetus. There are certain key cells of the placenta that allow this to occur, which include cytotrophoblasts and endothelial cells, for instance. One of the main cells of the placenta are the syncytiotrophoblasts, which are responsible for forming the outer walls of the placenta. These cells contain large portions of ERV proteins, specifically ENV proteins. The ENV gene in endogenic retrovirus codes for the protein in the viral envelope. If you think back to what we discussed earlier, the viral envelope is what allows the virus to fuse with the cell that's going to infect. Now, it seems strange that this gene would still be in the open reading frame of such random cells such as placental cells. However, what makes sense about this is that these placental cells have a similar function. The cytotrophoblast cells differentiate and fuse to form syncytiotrophoblasts. The ERVs are essential for this process, and without them, the placenta would not be able to have the same function it does today. The branch of scientific study is still developing. The negatives of ERVs are not fully understood yet. However, ERVs are believed to influence some neurodegenerative diseases such as ALS and MS. In ALS patients, HERV-K is expressed in much higher quantities than normal, which may affect motor structure inhibiting function. Well, I hope you learned a little bit more about viruses today.